And because the answer to that, so as you said, like every kid keeps asking why. So with all of these questions, physics too, if you start asking questions about, say, so why do fermions do this or that? It's because this mathematical object has a certain symmetry, and there's two kinds, and all of these things eventually end up in the math department. If you really want insight into what's going on, you end up in explanations that are not biology, they're not physics, they're properties of mathematical patterns. And if the mathematical patterns were different, the physics would be different, but also the biology would be different. In other words, cicadas would be coming out at different years if the prime numbers were differently organized. So we have to start thinking that some of the reasons for why certain patterns are the way they are not evolution reasons, and they're not physics reasons, they're actually math reasons. And so here's where the rubber really hits the road. We have been making creatures that have never existed before. So these are, as have other people, but we've made Xenobots, Anthrobots, large-scale systems that have never had a history of selection for this embodiment. The cells come from other creatures, but there's never been selection to be a good Xenobot or a good Anthrobot. If you want, I can tell you what some of them do. But the bottom line is they have very specific and very unique transcriptomes of physiological states, behaviors, shapes, these things, all of these things, very specific, very new.
Let's be concrete for people. What are you literally talking about right now? Like when you say Xenobot, what have you done in the lab?
Okay, so you isolate some cells from the epithelia of a frog embryo, okay, and you set them aside. You don't touch the genome, so no synthetic circuits. No,
you're taking cells from an existing creature.
You're taking cells from an existing creature. No scaffolds, no weird nanomaterials. You set them aside, and you let them reboot their multicellularity. They could do many things. They could crawl away from each other. They could die. They could form a two-dimensional monolayer like cell culture. Instead, what they do is they come together into a little spherical-looking thing. They have cilia on the outside. They coordinate the beating of those cilia so that the thing can start moving through water. They have various behaviors, and then they can do all kinds of interesting things. For example, they have over 600 differentially expressed genes compared to what they were doing back when they were inside the embryo. And remember, the only thing we've done them is free them from the bullying by other cells that make them to be like this boring two-dimensional cell, skin surface. Taking them away from those other cells reveals that by themselves turn on hundreds of new genes. Some of those genes are responsible for hearing and vibration, sound processing. And in fact, we found out that yes, unlike embryos, Xenobots do respond to sound that we provide with a speaker under the dish. They also do this crazy thing called kinematic self-replication. So if you sprinkle a bunch of loose epithelial cells in their environment, they run around, they collect those cells into little balls. Those little balls mature to be the next generation of Xenobots. And guess what they do? They run around doing the same thing. So you have generation after generation after generation.
Basically, you isolate some cells, some boring cells from an existing creature, and they will essentially reprogram themselves and behave like a new type of creature.
Yes, to be very clear, I'm not saying the cells were boring. I'm saying the other cells
I know.
We're confining them to a boring fate.
Yeah. They had all of this potential inside of them.
Yeah. And the human version of this, so let me just tell you about the human version of this, because it's completely wild. At that, when we made Xenobots and we were saying, like, look, there's never been selection to do kinematic self-replication or to have these genes. Like, there's never been any Xenobots. Where's this coming from? And people said, well, they're amphibian and they're embryonic, so maybe there's a plasticity. I said, fine. What's the furthest you can get from an embryonic frog? Well, how about an adult human? So what we did was we took tracheal epithelial cells donated by adult human donors for biopsies. Same thing. No genetic changes, no scaffolds, no nanomaterials. We let them self-assemble. They self-assemble into a little sphere.